Hello and good evening. Good evening to everybody. Can you all hear me clearly? Good evening to everyone on YouTube Live and Facebook Live. Please confirm if you can hear me and now you can see me, and you will be able to see the PowerPoint also now. Good evening, good evening. So, am I audible? Okay, fantastic. So, welcome to Biotechnica, everyone. Welcome to Biotechnica's yet another very powerful webinar on DBT JRF BET Biotechnology Entrance Exam webinar. And this evening, I am joined by two of the most eminent colleagues of Biotechnica, and they are going to talk in detail about this particular exam, DBT JRF. So before we get started, if I could quickly, get to know you in chat so that I could customize the webinar and our speakers could customize the webinar. So quickly in chat, if you can tell me, what's your qualification and which part of India you are from. Just type in the qualification like MSc Biotechnology and the city, say Bangalore, right? So just quickly type that in chat so that we know each other better. Okay, all right. Fantastic. So I can see a lot of people from all across the country. Okay, we have people from as far as Guwahati also. Fantastic. So welcome everybody to Biotechnica's special broadcast, special live session for all of you. I'm really thankful to each one of you for joining us today. You could have been doing something else which is important, but today, since you chose that you're going to attend this webinar, let me tell you that you are one of the action takers. You are the action takers, and that's very important, because once you have decided to do something, you're already halfway there. Okay, so all the action takers, are we ready? Shall we get started with the webinar? Shall we? Okay, yes, fantastic. So if you all want me to start the webinar, please type in the comment box BIO, bring it on. Let's see how many of you are ready. Are you ready with your pen and paper, notebook, whatever, to take notes? Okay, fantastic. So let's get started. Now that I have all of your permission, let me welcome all of you to this power-packed webinar, and I'm going to tell you why exactly you should be attending this webinar. Okay, so quickly, there are multiple reasons actually why you should be attending this webinar, but the first one being, it is only for those who want to secure a good future. If you really want to secure a good future, then you have a couple of things which you should be knowing. The first is, where do you invest your time on? Okay, so if you are going to invest your time on things, learning, unlearning and relearning things, that is what makes you a champion of tomorrow. And if that has to happen, this is where we get started. So this webinar is for those who want to secure a very good future. I want you to take smart steps. Okay, I want you to take smart steps towards this entrance examination while you are also studying for CSIR NET examination. So that is what the second point is. You have to take, we will tell you what smart steps you have to take in the wake of this uncertain CSIR NET dates. So the dates, till now we don't have a clarity, right? So what may happen, may not happen, but one thing we are very sure, the plus 2 exams have been postponed, CBSE exams are postponed, even the NTA has postponed the G and IIT and various NEET exams. So there is a highly likely chance that yes, of course, CSIR NET is getting postponed. So if that is the case, then why you want to base your future on something which is not certain? So we don't know CSIR NET is going to happen on the same dates or it is going to change. So why to take risk? Okay, there is a famous saying that do not put all your eggs in the same basket, right? Because if that basket gets spoiled, over. And that is what I want you to do is this year you must be writing DBT, you must be writing ICMR exam along with your CSIR NET, because maybe CSIR NET gets postponed, but all those things, all those concepts are already fresh in your mind. And if that is the case, why not write this exam, right? And that is what is my third point. You want to hit two birds with the same stone. Anyways, you are preparing for the exam. So what is the harm in writing one more exam? Just fill in the form. 1,000 rupees is the cost of the application, I guess. So go and write this exam, because if you are writing this exam, it is a pre-practice to the CSIR NET, of course, but you also get exam conditions, okay? So your mind gets conditioned to how exactly an exam is. So this is especially for those who are going to take CSIR NET for the first time. So now that I've told you 3 good reasons, the 4th one exactly is what I told you just now. If you want real practice, for DBT JRF and CSIR NET, you must be attending this webinar and you must be writing CSIR NET along with DBT JRF. There is no harm in that. Now that I have told you all the possible reasons, and I told you a very important saying, which is do not keep all your eggs in the same basket, okay? That means while you are preparing for CSIR NET, there are some overlapping topics, okay? So these overlapping topics is what we are going to talk today, and how you can strategize smartly so that while you are preparing for any other entrance exam, this also gets prepared, and then you give the exam, because the competition is much lesser. You can imagine, like, if 1 lakh people write CSIR NET exam, hardly 10,000 people will write DBT exam. So it's 10 times, okay? 10 times less competition. So your chances of selection goes up by 10 times, okay? So why not write this exam, and why not attend this webinar till the end? Now I'm going to give you one very special reason for attending this webinar till the end. And that reason will come after this slide. So stay tuned. Now, there will be obstacles. Coronavirus is one such obstacles. There will be doubters, people who will doubt on your capacity, people who will doubt that whether you will qualify or not. They can be relatives, they can be friends, or they can be anybody. It can be your own self. Self-doubt can happen, that whether I can write the exam. So there will be doubts, there will be obstacles, there will be mistakes. Nobody is denying that you're going to not going to commit some mistake. But with those mistakes, the more you write these exams, you get used to it, you learn from it, and that is what is the key to your success. So if you are ready for a hard work, there is no limit, okay? Like you've seen this ant, you can see that, right? So this is a saying not from me, it is from Michael Phelps. You know Michael Phelps. If you don't know, please Google it out. You will know how successful he was, because he was open to hard work. I want you to be open to hard work, and I want you to be using the smart tools and techniques which you're going to tell you today, and then you will be able to succeed. Your ticket to success, of course, is hard work, but slightly smarter way of doing hard work. Everybody does hard work, okay? It's like somebody takes the stairs and somebody takes the escalator, while somebody takes the lift. Now, which one you want to choose? Right. Now, in today's webinar, we are going to tell you the smart techniques and smart ways of preparing for DBT exam, but we are also requesting you that do not shun the hard work, okay? Hard work is also the key to success. So hard work, a combination of hard work and smart work, is going to be your key to success. Now, today's experts are going to be Dr. Tanushree Saxena, a senior DBT BET expert at Biotechnica. In fact, she has also designed our DBT reviser course, okay, DBT quick reviser course, which we call it. So Dr. Tanushree Saxena and Mrs. Sangrita Pal, DBT JRF tips and tricks senior experts she is, and these two experts who will join me just after this, I'm going to talk in detail about some very crucial things. Okay, so what we thought when we planned this webinar, we thought that many of you are not well versed with how, to apply, who is eligible eligible, who is not eligible. I can see a lot of chats coming in there asking whether I'm eligible or not. So what I want to do now is I'm going to tell you who is going to talk what. Okay, so the first speaker, who is going to be Tanushree, so she is going to talk about the basics. Okay, she's going to talk about how to apply the eligibility and the important dates. So while she is talking all that, listen to her very carefully, because there are some fine prints, and that is what I told you in my previous video on YouTube, where I told you that where I invited all of you personally. So that is where I told you that earlier you were at a disadvantage because only biotech students in their final year and just one year after the final year could give the exam. But now all those gates are open to all of you. So if all the gates are open for all of you, what is the harm? And very less people know about it, right? So if very less people know about it, if you apply for this exam, your chances of succeeding goes skyrocket. Now let's talk, and she'll also be talking about the basic intro to the syllabus. Okay, she'll not go into the syllabus in detail. She'll talk about the basic introduction, and she'll also share the exam pattern with you, so that before the second speaker comes, you are clear with what is the basics, how to apply, what are the last dates. All those things will be discussed. Now comes the second speaker. So second speaker, Mrs. Sangrita, will be talking about advanced tips, tricks, strategies, and she will also be sharing how this exam is similar to CSIR NET and GATE and what are the overlapping syllabus. Okay, and towards the end of the webinar, you will know with a very clear vision on what is to be studied for DBT, what we should leave, and how we should strategize. And that is what she's going to talk. And also she's going to talk about how to ensure guaranteed success in DBT BET exam. So both the experts of today are DBT JRF qualified, and they have extensive teaching and counseling experience for students like you. And now that they're going to join you live, the only thing which they expect, and I personally expect, is please be eager to learn. Okay, you must be eager to learn. So I want to know, are you all eager to learn? If that is the case, please type ETL in your comment box right now. Are you eager to learn? Please type ETL in the comment box. Okay, right. Yes, so I can see a lot of ETL coming. That means you're all power pumped up to learn, right? And that's what we want. We don't want to cater to students who get bored easily. We want to cater to students who are eager to learn, who are ready to unlearn and learn new things and implement it. Because, my dear friends, your success is our achievement. If we just tell you the tips and tricks and you don't implement, then it is of no use, right? So what I want is you must be attentive for the next 1.5 hour, roughly 1.5 hour, and we will try our best to answer all your queries in chat while our presenters are talking. However, we do not ensure that we will be able to answer to each one of you, but we will try our best. There are 6 experts only for you right now in chat. So whatever is your doubt, go ahead and ask while Mrs. Somrita and the other speaker, Mrs. Tanushree Saxena, is talking. Okay, so let's get started now that I have a permission. Now, before I pass on the controls to my colleagues, I have a special announcement to make. So we are going to give a surprise gift to all of you, whoever is attending this webinar till the end. Okay, now I'm going to be very strict. I'm not going to give to everybody who registered for the webinar. It will go to only those who will attend the webinar till the end and will submit the feedback form. So at the end, I will send you the feedback form in the chat box. Okay, our colleague will ping you there. You have to fill the feedback form, and only those people will get this surprise gift. Is that a deal? So are you going to attend till the end? If you are ready, then only I can start. Okay, fantastic. So I can see a lot of people are promising me that they will be there till the end. Thank you. And now that I'm going to pass on the controls to the first speaker. Over to you.
Hello everyone. Am I audible? Is my voice loud and clear? Please confirm. Okay, thank you. DBT BET 2020. I know most of you miss out this exam as you lack awareness about this exam, or even if you know, there is a lot of confusion among the aspirants. Like what is the mode of selection, mode of application, eligibility, exam pattern, syllabus, and most importantly, why one should give this exam. So are you all excited and energetic to hear all details about DBT BET 2020? If yes, please write "bring it on" in your chat box. Okay, I can see so many BIO coming. Fantabulous. As you know, Indian government departments like DBT has initiated several research programs to encourage students to pursue research in the field of biotechnology and life sciences in the country. But the question arises, what is the importance of research in life science? So here is the answer to this question: coronavirus. The highly contagious coronavirus outbreak started in Chinese city Wuhan and has then spread throughout the world, affecting several countries and endangered millions of lives. Currently, there are no specific vaccines or proper treatment procedures available for COVID-19. The entire country is under lockdown as self-isolation is the only prevention. And not only COVID-19. In the past decade, we must not forget that in the past decade also, so many viruses such as H1N1, Ebola, etc. have came into existence and challenged even the greatest of scientists, researchers, and doctors in the world. However, this has not stopped researchers and scientists towards finding solution for such catastrophic viruses. The corona pandemic has really unveiled the true importance of the biotechnologists. We all are hoping that very soon they will come up with something that will put an end to this global suffering. COVID-19 pandemic has made us realize the importance of research. Importance of research and has also ignited a global drive in this field to find solutions for the current situation. In future, there is going to be a need of young and dynamic minds in the field of research, which can contribute in welfare of society and mankind. So if you also dream of becoming a researcher, then it is the right time to fulfill your dream in the field of research, as the world needs you. Now the question is, how to achieve this dream? So here is the answer. DBT BET 2020 exam. DBT under Ministry of Science and Technology, Government of India provides fellowship to meritorious candidates for science pursuing research in frontier areas of biotechnology and life sciences through DBT JRF program. Junior Research Fellowship Program is the flagship and most prestigious program of Department of Biotechnology. It was initiated in year 2004 with one aim: to promote students to pursue research in field of biotechnology in the country. This program was initially implemented by University of Pune and of late Biotech Consortium India Limited is helping DBT with the implementation of this DBT JRF program. So this is about DBT JRF program. So what is BET then? Entrance exam name is BET, Biotechnology Eligibility Test. It is a national level exam which is conducted once in a year. Okay, please note this, once in a year. Unlike CSIR UGC NET where exam is conducted every 6 months, that is 2 times a year. So here you will get one chance in a year to give this exam. So I hope now it is clear what is DBT JRF and what is BET. Now let me take you through the mode of selection. 2 things to note here. BET is the computer-based exam. Selection for DBT JRF is through an online MCQ-based entrance test called as Biotechnology Eligibility Test, conducted at around 40 centers across the country. DBT JRF has 2 categories of fellowships. So based on the performance in BET, 2 categories of merit list will be prepared, Category 1 and Category 2. So let's find out what is this Category 1 and Category 2. The first category, it offers 275 fellowships. Students selected under Category 1 will be eligible to avail fellowship under the DBT JRF program. And fellowship will be tenable at any research institute or university in India, where selected candidates they can register for PhD. However, in second category, a merit list of 100 students is published. So the students here, they are eligible to join any DBT funded external projects in the country. DBT does not provide fellowship to these candidates. They have to avail the fellowship from the project that they are working in. And this fellowship is equivalent to other prestigious fellowships like NET and GATE. So if you crack this exam, you can work in any research project in research institutes of your choice. However, you have to pass through the selection procedure. Selection procedure like interview that will be conducted by the research institute of your choice. So this is an advantage for you because you might have come across the advertisements, research institute advertisements of research positions in their project. So they clearly mention that only DBT JRF candidates can apply. So this cuts out the competition because if anyone who has not qualified DBT JRF, those candidates are not eligible to apply. So category 2 is distinct from category 1 where the student has to mandatory register for PhD in order to avail the fellowship. However, in category 2, one has to join any project, research project, DBT sponsored project to avail the fellowship from the project. Now let's talk about mode of application. So this is the link where you will find your application form. So you can go through this link and there you have the complete the procedure, how to fill the form is mentioned, and you can fill the form easily. Now coming to the fees. So the fees is different for different categories. It is about ₹1,000 for general OBC EWS candidates, and for SC ST and differentially abled it is about ₹250. Now let's talk about eligibility. Biotechnology postgraduate students like MTech, MSc, they are eligible to write this exam. And even veterinary sciences, masters in veterinary sciences or equivalent degree, they are also eligible to write this exam. However, not only postgraduates, but those who have done 4-year degree courses after 10+2, they are also eligible to write this exam. Like BE, BTech, those who have done integrated BSMS, they are eligible to apply. Also, I saw someone asking in chat about pharmacy candidates. Pharmacy candidates are eligible to apply or not. So, pharmaceutical biotechnology candidates can apply. However, they have not mentioned anything about the pharmacy. However, those who have done masters in bioinformatics and computational biology, MSc or MTech bioinformatics and computational biology students, they can apply. And also those who are admitted under DBT supported postgraduate teaching programs, they are also eligible. And those who have done MSc in basic sciences, zoology, botany, they are also eligible, and even microbiology students, microbiology and biochemistry, biophysics, or though anyone who has done masters in allied areas of biology, life sciences, they are also eligible to apply. And not only the candidates that are pass outs, even those who are in their final year of their courses, okay, they are also eligible to apply. So this exam is not only for the pass outs, but those who are in the final year, last year of their course, they are eligible to apply. Now let's find out about marks. So postgraduate candidates, those who have secured 60% marks, they are eligible to apply. However, this much percent is for general EWS and OBC category candidates. For SC, ST and differentially abled candidates, it is about 55%. Now coming to the age limit. So anyone below 28 years or up to 28 years as on the last date of application, they are eligible to apply. However, this age limit is for general and EWS category candidates, and relaxation is given to some candidates, like it is about 5 years. 5 years relaxation is given for SC, ST, differentially abled and women candidates, and the relaxation of up to 3 years, that is 28 plus 3, that is 31 years, it is for OBC and non-creamy layer candidates, relaxation is given. Now coming to important dates of BET 2020, DBT BET 2020. So as you can see here, registration has already started. So mode of registration is only online, no offline mode is there. And so I have already mentioned the link. You can go through that link and fill the registration form. And those students who are in the final year, they can fill in result awaited category. Okay, like in the previous slide I mentioned that 60% marks is required. So obviously those who are in the final year, they can fill the form in result awaited category. Then the registration is closing on May 18. So still you have time. So those who have not registered, they can register. And this is the exam date, that is June 30th. So this is the tentative exam date because we don't know that whether the date will be extended because of COVID-19. So for now we stick to this date. And please note, okay, I want you all to note this date, to save this date, as you all have to attend the exam. You should remember this date. And please don't wait for extension, because DBT and BCIL, they are very particular. So try to fill before registration closes. Then you can see that the display of question paper and answer key on the website will be on the same date. And the declaration of the result will be on July 28th. And you know, even after qualifying this exam, you can activate the fellowship within 2 years of issue of award letter. Now coming to why to give this exam. What is so special about this exam? Why you should give this exam? So the first reason is that this particular exam is inclusive of all life sciences. So if you have done MSc in biotechnology or MTech biotechnology or any such degree, you must write it, because it is exclusive for biotechnology candidates, and also it includes It includes some of the few sections of the life science industry as well. So as only few sections they are involved, they are allowed, so that means competition is lesser. So definitely it is the benefit for the students, those who write this exam. Then another reason is awareness about this exam. Since very few people know about this exam and write the exam, hence the competition is lesser. This is also a major reason for a higher success rate in this exam compared to others. So take this to your advantage. Okay? Then write the exam while you are studying. This is another advantage. This is the right time to attempt to this exam while you are still studying. So it is must for all biosciences candidates who are pursuing their masters in biotechnology or bioinformatics and others that I have mentioned. You need to give this exam. Then another reason is directly get into PhD after masters. So this is another benefit of writing this exam, because as soon as you complete your masters and if you have qualified this exam, you will be directly absorbed into the fellowship and you can start pursuing your PhD immediately with no career break or gap. However, you still have an option to activate your fellowship within 2 years. As I told you earlier also, you have the option to activate your fellowship within 2 years of the issue date of the award letter. Another reason is syllabus is comparatively easier. The syllabus of DBT JRF exam is much easier compared to other exams in biotechnology, because most of the syllabus is covered during your masters. I'll be discussing about this syllabus. So you can always plan taking up other exams like CSIR NET, GATE, ICMR as well. So if you are doing so, you are preparing for the DBT JRF exam as well, because syllabus of almost all other major biotech entrance exam gets covered, right? If you are preparing for like CSIR NET exam, so the syllabus of almost all other major biotech entrance exam gets covered. So that is why it's an opportunity. These are the reasons why one should give this exam. Now let's talk about exam pattern. The duration of the exam is 3 hours, that is 180 minutes. And marking scheme, that is 3 marks for every right answer will be given. And no negative marking is there. So this is again an advantage, because most of the other competitive exams, they have negative marking. So here you don't have any negative marking. The test contains 200 questions, which are divided into part A, that is section A, and section B. So in part A, section A, it comprises of 50 questions and all of which are mandatory. Okay, please note, all of which are mandatory. However, in part B, you have to do only 50 questions. You have to attempt 50 questions out of 150. So like if you see CSIR UGC NET exam, so there you have a choice, like in part A, out of 20 you have to do only 15, in part B out of 50 you have to do only 35. But however, here this is not the case. In section A, in part A, you have to attempt all the question. Now one important thing to note here is that if you do not do all the 50 questions, it doesn't mean that your paper will be rejected. No, it's not the case. However, it is, you should try to solve all the 50 questions as negative marking is not there. Because sometimes what happens in some competitive exams where negative marking is there, you know the answer, but just simply because you are not sure, you leave that question, right? You don't attempt that question. But here, as the negative marking is not there, you can attempt the question. So overall total marks, it is of 300, because as I told you that 3 marks is given for every right answer. So if the marks for the right answers, the marks for these 50 will be included. So overall the total marks is 300. Now let's talk about the syllabus. So as I told you that you have 2 parts, that is part A, which is all about basics, and part B, that is about specialization. So let's find out what is included in part A and part B. Part B is about aptitude. So part A, it basically involves two topics, aptitude and general biotechnology. So in aptitude, you have the questions that include comprehension related, where the candidates, they are given a written paragraph to read and then questions related on that paragraph. So they might be designed to test the non-verbal reasoning capacity, and they may also be quantitative in nature, designed to assess candidates' ability to comprehend large numbers and to perform basic calculations. So these are not very difficult. These are basically of 10+2 level, so they are very easy to do. However, if we talk about general biotechnology, so in general biotechnology you have, general biotechnology involves several topics, and in general biotechnology this is comparatively easier because this is something that you are already studying. This is not new to you. You already know about the structure of lipids, proteins, you know conformation of nucleic acids, you know stabilizing interactions, because most of the syllabus is already covered in your masters. You know about transcription, translation, replication. This is something you are already studying. About the genetics, right? However, there are certain topics like techniques, microarray, PCR, sequencing techniques. So there are several techniques which can be new to you. So what you have to do is, the strategy for preparing for this part A is that strengthen the topics that you already know, and those which are new to you, you can focus on them. And please practice as many questions as you can. So you will find the previous year question papers in DBT at DBT and BCL website. So you can download the previous year question papers from there with the answer keys given. So go through this. This is something that you are already studying. So I emphasize that please focus on general biotechnology, as you have to answer all the 50 questions, right? Then when we talk about part B, so here is this part involves the specialized branches in biotechnology. So here you have agricultural biotechnology, where you have the questions related to molecular biology, seed technology, plant biology, tissue culture. Then you have molecular breeding and genomics, abiotic and biotic stresses, biodiversity. Okay. Then animal biotechnology, where you have the questions based on animal physiology and biochemistry, histology and embryology, animal parasitology, animal virology, immunology, animal genetics, and so on. Then another unit is about industrial biotechnology. Now this is very important, okay, because as DBT is conducting, so they emphasize more on industrial biotechnology based questions. So this is very important from exam point of view. It's mandatory, like if you do this unit, definitely you'll able to attempt many questions. So industrial biotechnology is all about engineering principle, thermodynamics, bioprocess engineering, downstream processing, and also bioprocess plant design. Then another unit is bioinformatics and computational biology. So here you have all the syllabus, all the topics related to bioinformatics and computational biology, like scoring matrix, sequence analysis, database searches, and how drug designing, that is all included. Then environmental biotechnology, here this unit is very easy because it is all about pollution, water pollution, and also basic ecological concepts and principle, and management of the waste, municipal, biomedical, and agricultural waste, environmental pollution control, environmental friendly technology, pollution monitoring, and global environmental problems. Then in marine biotechnology, it is all about marine organisms and their behavior, marine resources assessment, population study, marine environmental protection, oceanography, microbial assessment, types of marine microbes, and what is the role of these microbes, marine microbes, role of microbes in marine environment, microbial interaction. Okay, so this actually, among all the subunits, this is very vast, this particular marine biotechnology. So many topics, subunits are included. And then you have medical biotechnology, which is all about vaccinology, immunotechnology, genetics, genetics of neurogenetic disorders, eye disorders, comparison of genetic counseling, infections of gastrointestinal tract, respiratory systems. Okay. Then you have host pathogen interaction disease process, gene therapy. Again, it is so many topics are included here. Human disease gene silencing technology. So medical biotechnology is again another unit which is very important. And then you have the molecular and human genetics. This is again important, where you have the principle of genetics, cell biology and cytogenetics, then genetic disorders, human genome. Okay. So if you see that, if you compare part A and part B, 25 to 30 questions, they are common, right? That means if you because genetics and about these disorders and all, you're again studying in part A. So that means if you prepare part A thoroughly, general biotechnology part thoroughly, you can easily do the questions from part B as well, right? So please make sure that you emphasize on general biotechnology part A topic very well. And also, so most of the topics from part B will be covered. And as I told you, some of the important units like industrial biotechnology, medical biotechnology, to please focus on them. Then another unit is neuroscience. So neuroscience is all about neurons, glial cell, neurophysiology and behavior. Okay, birth and migration of neurons, neurotrophic factors, and also about the sensation and perception, synaptic transmission, fundamental of motor systems. So again, it is based on chemical control of brain and behavior, and sleep and dreaming, cognitive development and aging. So that is all included in neurosciences and much more. Then pharmaceutical biotechnology, it is all about drug metabolism and enzymes and microbial technology, biochemical engineering, downstream processing and industrial application. So if you are wondering that the topic, the syllabus is so vast, how you're going to prepare for it, so the strategy is smart study. Okay, focus on 3 to 4 units. Now, what 3 to 4 units? The units that are important from exam point of view, and also the unit of your interest. Like some have interest in agriculture or animal biotechnologies. So accordingly, you can choose your unit. And as I told you, like 10 to 15 questions you can expect from industrial biotechnology. This is an important unit. So don't leave this unit and focus on general biotechnology. Part A, general biotechnology. So you're able to attempt many questions, right? And practice as many questions as you can. So you will find the previous year question papers in DBT and BCIL website. So you can download the previous year question papers from there. You can see the pattern, and even you will find the answer keys there. So practice many questions as you can. Okay, and more details about the syllabus will be discussed in next session as well. So you'll find more about the syllabus there. Now, if we do the analysis of syllabus, so syllabus is very diversified. It is very vast. It includes not only agriculture, but agricultural biotechnology, but also animal biotechnology, medical biotechnology, industrial biotechnology, and many more. So the area of interest, like it is all about your aptitude and intelligence. Like area of interest, it depicts your aptitude, but it doesn't guarantee you to excel in that area unless and until you are intelligent enough to work hard and make that aptitude your strength. Okay, so the syllabus, you have to study, you have to select the units based on your aptitude and also intelligently. Then syllabus is again very classical and applied. That is, not only you will be studying the very basics of very basic things like about the structure, about the interactions, but again you will have the application part there. Like if you are studying about any technique, right? If you are studying about PCR, if you are studying about microarray, so not only the principle of that technique, but what is the application of that technique for the benefit of mankind, that also you have to, the emphasis need to be given. And some topics are very easy, simply you have to remember the terms, definitions, but for some, in order to understand like some techniques. Not only you have to remember the principle, but the application part as well. So the success strategy is that start from basics to specialization. So if you start from basic, what you are already studying, and you just focus on the points that you already know, if you work to strengthen the points that you already know, and so that will definitely help, and then you can move on to the points that are new to you. You can focus on them. So the key to success is start from basics and moves to specialization. So start preparing now. You still have time, and most importantly, believe in yourself. I know you all can do. Everything is possible. Remember, everything is possible if you believe. Okay? So all the very best. And now I request my colleague, Mrs. Sangrita, to guide you further with the preparation for this exam.
Thank you, Dr. Tanushri, and good evening, students. So I presume by now you have developed a very clear idea about this DBT examination, its structure, the question pattern, right? So taking this discussion forward, I will be sharing with you some more insight regarding this examination. And in my discussion, I will be talking about the DBT syllabus and how it is close to your CSIR or GATE syllabus. Now see, when you are preparing for DBT, okay, you are basically preparing for what? To do some PhD work, right? To do your research. That must be your target. That is your dream. All right. Now, when you are preparing for one single exam, if you just analyze the syllabus of all the related exams, okay, all the exams which are there for a research purpose, right? Your DBT, your CSIR, the GATE, the ICAR, okay, ICMR, all of these exams. If you analyze their syllabus, you will find something is common in all the syllabus. All right. So my suggestion is always that when you are preparing for one exam, why not prepare for something which is, similar? So simultaneously you prepare for other exams as well. So if you are mainly targeting for CSIR, all right, because of course CSIR is very important. Why? Because that gives you a lecturership in college as well. So after your PhD, after the research, maybe after even your postdoc, if you want to get into a college, you have to have the CSIR certificate, right? So if you're targeting for CSIR, target for other exams as well. And as ma'am's already mentioned, that DBT has got a very high opportunity for you, right? Because here the competition is less than that of CSIR NET, all right? Yes, Pooja, I will be going into the details. I will show you the similarity between the syllabuses. Okay, so just keep following. I'll just come to it. Okay. Now, so here I'm going to mention some easy way out for you people. Okay, because when you look at the syllabus, I guess many of you feel quite nervous. You think that, okay, this is such a big syllabus, whether I'll be able to complete it or not. Alright, you don't know from where to start and where to end. So here I am to give you some tips and tricks regarding that. And I will also tell you how Biotechnica is going to help you, okay, in your preparation, because all we want is your success. We all want the shining stars. So are you all eager to learn more about the syllabus and see how this difficult syllabus can be made easy? If you're eager, then you can type ETL. Yes, right. So I see a lot of ETLs. Okay. No, Rahul, your DBT doesn't give you the permission to sit for the College Service Commissions, okay? But it does give you the fellowship to conduct your research. Now see, even when you are appearing for any interview in the college for the post of your assistant professor, you will find most of the candidates, they are already having a doctorate, okay, or even some of them are having postdoctorate certificates, okay. So it's always better for you to first complete your PhD. All right. So now your target should be qualifying any of these exams to fulfill your dream. I'm sure all of you have a dream of doing research work, but obviously your choices will be different. Okay. So let's come back to this topic here. So the first thing that we are going to discuss is the DBT syllabus. Now you already have seen the breakup of the syllabus, right? The syllabus, you have it divided into part A and part B. Part A is consisting of the general aptitude section and obviously the general biotechnology section, okay. Whereas part B, this is more focused on specialized section. So depending upon what is your specialization, you will be selecting the topics from your part B. So part A consists of 8 subunits, all right, under your general biotechnology section. Whereas part B has got 10 subunits, okay. Now, in your part A, you are required to answer 50 questions out of the 50 questions which are provided to you, all right. But it is not like that if you don't attempt any question, your paper will be disqualified. Not like that, okay. Here the options are not given. Like it is not given as you can answer 3 questions out of the 5 questions. You are not getting that option, okay. All the 50 questions you are asked to attempt those 50 questions. So if you don't know those 50 questions or if you don't know 1 or 2 of these questions, you are free to leave it, okay. Whereas in your part B, you are getting the option. So out of your 150 questions, you are only required to give 50 questions. Okay, so you see, here you are getting so much of an option. Okay, and trust me, your part B is actually game changer. Okay, it can actually make you crack this DBT exam. I will tell you how. But before that, let us just go through this syllabus. All right. Yes, Shruti, I'm just getting into the details of it. Okay. So while I'm going to discuss this syllabus, I will not only show you the comparison with the other exams, but I will also tell you what are the important topics that you have to study from each of these subtopics, okay, or the subparts. So you can be ready with your pen and paper. So while we are discussing, you can jot those points down. Okay. So are you ready, all of you? Yes? Yes? Okay, fine. So see, I'm basically going to talk about the general biotechnology part. Okay. So the first subtopic you have here is your biomolecular structure and function. Now this section is related to your biochemistry. Alright. Biochem section. So if it is a biochemistry section, then what are the important topics? You will be asked questions from the proteins, lipids, carbohydrates, okay, nucleic acids. Then you have the metabolism, okay, then structure and function, the chemical kinetics, free energy, thermodynamics, okay, entropy, enthalpy, and very, very importantly, your enzyme kinetics. All right, enzyme kinetics is very important. So they ask a lot of questions from here, and they can ask some questions which are based on your Vmax, your Km, okay, then your specific activity of the enzymes. They can ask all of these things. All right, even they can ask you about the equation, okay, and you might get some graph based questions from here as well. Okay, and another section that is important under this is your photosynthesis, the nitrogen fixation, all of these. All right, so here you have a lot of things to memorize. Now, next part, that is your methods in biotechnology. So methods in biotechnology, it is dealing with all the techniques. All right. So all the technique based questions will be asked from here, like you have the chromatography, spectroscopy, gel filtration. Okay, these are all the important topics that you must learn from this section. Then you also have the electrophoresis, microarray, microscopy is very important. So then you have the in situ hybridization. All right. So when you're studying this technique, make sure you study the working principle properly. All right. You study the working principle and then you follow the paths. Okay, what is happening, why it is happening, what are the things which are used, why they are used. All right, those kind of questions you have to ask yourself why you are studying it. All right, because in your exam you will get these kinds of questions more. All right, so basically remember one thing when you're preparing, DBT is going to test your concept. All right, so that's why you have to have a very clear concept regarding all of these techniques. Okay, now the third point is your organization of structure and function of prokaryotic and eukaryotic cells. That means this is talking about your cell biology. All right, so cell biology means it is going to talk about your cell wall, cell membrane. You will get questions from the organelles, okay, their structure, their function, and most importantly, when you're studying the organelles, you have that protein sorting, okay, where your protein is being folded from where it is going, like from ER to Golgi, okay, and everything that is related to this protein sorting. Okay, and again another very important topic from here is obviously your cell cycle and cancer. Okay, you have the fourth topic as your cellular processes. Now cellular process means it is going to talk about your central dogma. Now all of you know about central dogma, don't you? So central dogma is that RNA, DNA, RNA and protein, right? So over here the questions will be based on these processes of replication, transcription, translation, okay, about the enzymes and their activities, okay, about the inhibitors. They can also ask you questions on codons, all right, codons. Then you can have the signaling pathway asked from here. And another important section which is there in the cellular process is obviously the immunology section, okay, where you have this antibody. So they can ask you about the antibody antigen reaction interaction, okay. They can ask you about this hybridoma technique. So you must learn about all of these, the activation of B cell, T cell, and specifically vaccine, because that is an applied part of your immunology section. Okay, so these are something which I will highlight in this fourth point. Now coming to the fifth one. Fifth one is dealing with your recombinant DNA technology. So recombinant DNA technology means it is going to deal with the enzymes, vectors, PCR applications. Okay, they will ask you questions about your gene library, gene cloning strategies, all of these. All right, then you have the genomics and proteomics. Now this is a very important section, not only from the point of view of your exam but also afterwards. So when you will be conducting your own research, okay, or you might be facing some interviews, mostly you will be requiring some basic knowledge from this genomic and proteomic section. All right, so when you're studying this, study it well, not only for the exam but for the later purposes also. And then you have this part 8, which is talking about your IPR, the biosafety and the bioethics. All right, so in this case, in this one, what you get question is from your trademarks, the copyrights, the patents. Okay, then the question might be asking what happens if you infringe the patents. What will be the legitimate thing if you infringe the patents, the scope of the patents. All of these will be asked from this section. Now coming to the part which you are waiting for, that is the comparison with the CSIR exam, GATE BT and GATE Excel. Yes, see, you can compare your DBT syllabus with 3 other related exams. All right, the main exams that you all aim for. So the first one, this is your biomolecular structure and function. And as I told you already, this is dealing with your biochemistry, right? So if it is dealing with your biochemistry, then in CSIR NET, this has got similarity with your Unit 1. So CSIR NET, do you remember what are the different units? Unit 1 is talking about the biochemistry. So while you are preparing for your CSIR, see, you are also preparing for the biochemistry from your DBT exam. If I talk about the GATE BT, it is under your section 2. You have biochemistry, a topic. Okay. So again, something similar. Then for your GATE Excel, now GATE Excel has got 5 papers, right? Of which you have one plant section, you have the the botany, the zoology, you have the microbiology, biochemistry, and obviously the nutrition. Okay, so in this case, biochemistry part is absolutely common over here, the biochemistry paper, but you will also find sections related to biochemistry in your botany part. The botany paper, if you opt for, you will also get it in your zoology. Okay, there are these sections mentioned there in that syllabus as well under botany and zoology. Now coming over to the second point, methods in biotechnology. Now methods in biotechnology, it has got a similarity with your Unit 13 of CSIR. Okay, and again you will get a common point from here under your GATE BT. Okay, biotechnology GATE under your section 2, where you have the head analytical techniques. Okay, the analytical techniques. All right. Again, some of the techniques which are mentioned under biotechnology, you will find some common techniques given in your zoology paper as well under your GATE Excel. Okay. The next you have is third one, organization of structure and function of prokaryotic cell. Now this is also a very common topic. So in your CSR exam, this topic is mentioned under Unit 2. Okay. In your GATE BT, this topic is again mentioned under section 2, cell biology. Okay, there is a different topic which is dealing with all these subtopics. Okay, you will find a mention of this again in your GATE Excel botany paper and zoology paper. All right. Now coming to your fourth one, cellular processes. Cellular processes, whatever is mentioned here, like you have the transcription, translation, those sections are in common with your Unit 3 of your CSIR, and the immunology section, okay, the signaling part, all of those are common with Unit 4. So there is a combination of Unit 4 and Unit 3 placed under this one single head. Okay, now this is also a very common topic. So you will find a reference of this in your GATE BT as well under section 2. All right, where you have this cell biology. Under that section only these cellular processes are mentioned. All right, and also you will find a reference of this under your GATE Excel botany and zoology paper. All right. Now coming to the fifth one, that is your recombinant DNA technology. Now recombinant DNA technology, the techniques which are mentioned here has been referred to Unit 13A of your CSIR paper, okay, very specifically. And you will find a mention of these techniques in your section 3 of your GATE BT question. Okay, section 3 is dealing with your recombinant DNA technology. All right, you just look for these heads, you will find similarity. All right. Now the seventh one. Seventh is genomics and proteomics. All right. Now genomics and proteomics, certain references you will get it in your CSIR Unit 30. Under this head, you will get some topics mentioned. All right. Whereas no such, topics are there or highlighted in your GATE BT or GATE Excel. Okay. Now this part 8 IPR, biosafety and bioethics. This is a very unique topic, all right, which is very specifically asked in your DBT exam. So see, over here, when you are preparing for your CSIR, okay, when you're preparing for your GATE, obviously when you're preparing for your CSIR, you are going to prepare about Unit 1, Unit 13, Unit 2, Unit 4, Unit 3. These are very, very important topics in your CSIR, and you just cannot miss these topics, right? So see, simultaneously, you're also preparing for all of these subtopics under your general biotechnology, right? Even if you're preparing for GATE BT, still you are getting some common topics under your DBT. Okay, so that is why I mentioned earlier that when you're preparing for one exam, why don't you sit for the other exams? Because actually when you're preparing for one, you are preparing for all the other exams. Alright, there will be slight differences, but not a huge one. And those slight differences, like this one, this topic, IPR, this is a slight difference and it is manageable. Okay, I will tell you how you can manage this, but it is manageable because a major section is being covered under a common preparation. Okay, so you must go for your DBT exam. Now the second part is your general plus specialized branches. So this is mainly dealing with the specialized section. Okay, so under this you have agricultural biotechnology, animal biotechnology, MTech, biotech, biochemical engineering and industrial biotechnology. You have bioinformatics and computational biology. Then you have environmental biotechnology, you have marine biotechnology, medical biotechnology, molecular and human genetics, neuroscience and pharmaceutical. Okay, so over here basically this agricultural biotechnology, this is for the botany people. Okay, so those who are from the botany background, you do attempt this paper. Okay, this is for you people, and this is also for the other candidates as well. Because see, no matter if you are coming from zoology background, microbiology background or any other backgrounds of your life science, when you're going to study for CSIR exam, you will be studying about the botany section as well, Unit 6, right? Unit 6, it is very common and you must study this. So if I again compare your first part of this special paper, agriculture biotechnology has a similarity with your Unit 6, and it also has got portions from your Unit 12, the tissue culture, the plant tissue culture processes. All right, and also it has dealing with your biodiversity and all, which comes under your Unit 9. So it is a combination of 6, 12 and 9, okay, from your CSIR exam. Now this section is also present in your GATE BT. Yes, it is also present in your GATE BT under section 4. So under section 4, you have got this plant and animal biotechnology section. Okay, that is dealing with a portion of your agricultural biotechnology and animal biotechnology. Okay, both together you have it. And of course, if you look at your GATE Excel, then you have this under your botany, the whole of botany. Okay, the classical botany part. Now, coming to the animal biotechnology. Animal biotechnology is dealing with the animals, that means the zoology part. Okay, so those who are from the zoology background, you have a very good scope of opting for this animal biotechnology. Alright, so if I again compare it with your CSIR, then your animal biotechnology is dealing with your Unit 7 and portions of Unit 4 as well. Alright, and obviously it has got a mention in your GATE Excel under the zoology paper. Alright, now this third point, this is very, very important in your DBT. Why is it important? Because over here you have maximum questions asked from this part. Okay, you have almost 10 to 15 questions which are set from this section, MTech, Biotech, okay, biomedical engineering and all. So this section has a very similarity with your GATE BT. So those who are from the GATE BT, who are preparing for the GATE BT, or you're from the biotechnology background, you can opt for this paper, okay, for this particular section. Coming to your bioinformatics and computational biology, this part is talking about the all the things that we deal about these days. All right, so this has got a reference in your GATE BT again under your section 2. Section 2, there is a subtopic named as bioinformatics itself. Okay, now environmental biotechnology, though the basics are related to your unit 10 of CSIR, but over here when you're preparing this, you have to focus on the technology sections. Technology sections as in what? You might have to know about the population, how you're measuring pollution, how you're measuring population, what are the parameters, okay, what is the current data that is available. So you have to focus on more of the applied sections over here than the theoretical part. All right. Marine biotechnology is a very unique topic again, which is very specific for your DBT. And next you have the medical biotechnology. Now medical biotechnology, this also has got some references to unit 8 because it is dealing with your the genetic sections, okay, the human genetics part. Okay, and it has got a reference to unit 13 as well. So the ELISA's techniques, okay, and you have the ECG techniques, all those techniques which are used in the analysis of the human diseases. Okay, those are mentioned under your biotechnology. So again, you are finding some similarity here. Now molecular and human genetics. Under this also you have some references from your unit 8 of CSIR and unit 4. Okay, and you also have a reference of this in your GATE BT under section 2. So in section 2 you have a topic named as immunology. So that topic comes under, okay, as a part of this subtopic molecular and human genetics. You have to study about immunology here as well. Okay, and of course there are certain biochemistry related questions, related topics, so that comes under your Excel biochem. All right, and again your neuroscience and pharmaceutical, these two are very unique topics specific for your DBT. All right, so can you see that if you are preparing for one exam, you are getting so much similar with the other, right? So now let us see what to do when we are getting this syllabus. Okay, so we have to start planning properly. Okay, we can't just start studying every part of the syllabus. Once we know what are common, what are the important questions which are asked, okay, we have to now sit and think first. We have to plan something. Okay, so now coming to the tips and tricks section. So remember, in order to qualify any competitive exam, hard work is not sufficient. Basically, more than hard work, you have to go for a smart work. Okay, there is a basic difference between hard and a smart work. A person who is working smartly will be sitting and planning first. Okay, and then he will be focusing his hard work on those topics. All right, rather than just take it blindly. So if you look at the distribution, which you already have seen, you have to answer 50 questions from part A. That means what? Total of 150 marks. All right, because there is 3 marks for each of these questions. Now part B has got, you have to answer 50 questions once again, okay, out of 150, and you have 3 marks here as well, and negative mark you are having 1. Okay, now so from part B also you can get 150 marks. So in total you are getting a question paper which is of 300 marks, right? 150 plus 150 will give you 300. Now if you look at the cutoffs in the previous years, you will find that 181 has been the highest cutoff so far, all right, under general category. So what I will tell you is try to get a score which is closer to your 200. Okay, then you can think yourself to be on the safe zone. Okay, now if you want to get a score of 200, you have to plan your study accordingly. Alright, so what I have shown you, the similarity and all, don't you think part 1 is easy to study, right? So part A, if you study it very well, okay, then how many you can score from here? Maybe all the 50 marks, 50 questions, alright? You are getting approx your 150 marks over here. Now, suppose you don't get 150, okay? On average, let us see, suppose you are getting 100, alright? Fine, we are taking 100 marks over here. What about part B? Part B, as I have mentioned here, you have to just select 3 to 4 subunits from here. Yes, my dear, you just have to select 3 to 4 subunits. Yes, Rahul. Yes, Prakash. Just 3 to 4 subunit is sufficient. Why? See, because you are getting approximately 5 to 6 questions from each part. Only the maximum section is coming from here, about 10 to 15. So I will insist that no matter how difficult it is, at least learn some things from this topic. Okay, don't leave it out completely. Alright, because you are getting the maximum questions from here, 10 to 15. Alright, the rest you can select from your specialization. So those who are from botany section, those who are strong in botany, go for agriculture biotechnology. In fact, I will tell about all the life science students who are currently studying for your CSIR. Since it is common with your unit 6, go for your agriculture biotechnology. Okay, those who are from zoology, definitely attempt animal biotech. Okay, you can look up certain things from this section, which is your bioinformatics and computational biology. Environmental biotechnology you can take up. Okay, medical biology and molecular biology you can take up. So what you can take is molecular human genetics, medical biotechnology, environmental biotechnology, and maybe any one from these two. Alright, so that gives you 3 to 4 topics, and I will suggest please go for this one as well, because you are getting more options here. Okay, now if you just compare this with your part A, you will find you will get almost approximately 25% common, or 25 marks common to your part A, because there also you have to study the techniques, there also you have to study the genetic parts, there also you have to study immunology. Okay, and those are there over here mentioned as subparts in your under these subtopics. Okay, so that is what you can do. So this is the first thing that you need to focus on. So after selecting, what you're doing, you have to plan properly. Okay, once you get the syllabus, then you have to learn those topics that you have selected. Then you have to train yourself to think. Okay, because many a times question can come in a twisty turny manner, and you have to think in that way. Alright, so you have to train your mind to think, because we are mostly habituated with note study. Okay, but that will not happen here. Fine. Next is practice. Of course, practice makes a man perfect. We all know about that. Okay, so whatever we have learned, however we have trained, we have to implement that. Okay, and we have to do it in a constant basis. Alright, we have to solve questions, we have to revise those topics that we have done. So all will come under your practice. And lastly will be your performance. So performance means what? How you are performing when you are getting into that examination hall. Only if you go in this pattern, then success will be very easy to you. It will be in your hand. Alright. So let's take up one at a time. So planning. So once you get your syllabus, alright, study the common topics first. So common topics will be the topics which I have highlighted, alright, in your part A section, like your biomolecular structure and function, the methods in biotechnology, the organization of structure and function, the cellular processes, recombinant DNA technology, genetics, phylogenetic evolution, all of these can be the common topics, even the genomics and the proteomics section. So you first study these parts, okay? And of course your agricultural biotechnology as well. Next, when you are studying, okay, divide the time slot. So unit time slots on rotation is very, very important. So what you do is you assign day slots for units in a week, all right, or assign hour slots for subunits in a day. So divide the 24 hours. Of course, when you're dividing, do keep time for sleep and eat, okay, and to relax as well. Relaxation is very important, okay. So as revision is important, relaxation is also important, all right. So keeping time for all those things, the rest of the time you divide into slots. Study not more than 2 units in a day, otherwise you will just become haphazard, okay. And preferably take up the linked units, okay. So when you are studying for your cell structure and function and all, you can take up with that the cellular processes, all right. With cellular processes you can link the cell signaling part, okay. Like that way you can link all the related topics, because that will help you to form a bigger picture, and bigger picture formation is very important when you're preparing for a exam, okay. Next, divide time slots for different activities, okay, under a topic. So like this way you can divide your time slot. First slot, study the topic and understand the concept well. Second is go through some questions from the topic from previous years and question and make notes of those subtopics, okay. Third slot, you can try to solve some questions from those subtopics, all right. And fourth, revise what you have studied the previous day, all right. This helps you to learning is your reading, and then it should be a compaction of what you have read, okay. So when you're revising, when you're solving the question, you are actually forming a concrete thing of that particular topic, okay. Then study timing in a day. So designated hours should be there for the study, all right, and minimum study 5 to 6 hours every day, okay. Now keep the difficult topics to be studied in the most effective and productive time of the day, which is that it is the morning time, okay, when you're fresh in mind. That is the most productive, I feel. But if you think that no, your productive time is late night, then of course you can keep those difficult topics in those timings also. And of course, take small breaks and power naps whenever you feel saturated, because that if you don't take breaks, it will stress your mind, and a stressed mind cannot study, okay. Even if you think that I have memorized this section, the next moment when you try to recall it, you will find that no, those sections are still not yet prepared. Yes, I know you, it happens with everyone. Yes, Prakash, it happens with everyone, see, okay. So you take breaks, okay. Next is learn. Learn means how you are preparing. So make notes. So when you're making notes, highlight the important points, okay, like the key terms, definitions, equations, units, theorems, all of these, highlight them, all right, so that they are easily, pointed out, all right. So try to visualize things in your imagination. So when you're studying about a process, like when you're studying about, say, replication, okay, try to imagine it. So you might be studying from an animation, but when you are recalling it, play that animation in your mind with all the informations that you have, because once you make that picture in your mind, it will be easier for you to recall, okay. Next is avoid photographs. Avoid stuffing too many information in one page. So keep space when you're making the notes, all right. And I will personally tell you to make flowcharts and tables when you're making the notes. Don't go on writing pages after pages. That doesn't help you, okay. So make flowcharts and tables. You can make flowcharts and tables on signaling, instrumentation, cellular processes, okay, enzymatic reaction, like this one that I have done for you. This is regarding your PCR, all right. Now PCR, the whole process, what I have done, I have simply spaced it out and made it under a flowchart. So see, this is easier to learn, this is easier to remember, okay. So you can make flowcharts like this. Then jot down the working principles and formulas. Like here again, this is your reference to PCR. See PCR, what I have done, when it is used, what are the limitations, what are the basic components. All of these things I have simply done it in a small place, right? Primers, see how I have written short nucleic acid segment hyphen 21 to 45 base in length hyphen one set of primers, okay, that is hyphen forward and reverse. So you don't have to write words, okay, just the points are sufficient for you to recall the whole topic. Now, charts and tables are very, very important, okay. So keep adding to the existing column. So when you're making a table, keep, adding whenever you are coming across a particular information. So that table you can make on your enzyme regulations, signaling pathway, drugs and uses, all of these, okay. So here is one example that I've provided here. So you can see there are tables and the tables are spaced out properly, okay. So keep this in hand, especially for immunology. This table will be very useful to you, okay, even for the hormones and signaling, these parts. Now, you can also have a printout files, okay. Printout files mainly is what they are self-explanatory, okay, in nature. So when you look at the printout files like this, when you have a colorful diagram, immediately all the information comes to your mind, okay. And when you're looking at a diagram, try to recall all the related things which are there along with this diagram, okay. So in a go, you will see that you have actually recalled the whole chapter, okay. Or you can take this cycle, okay. You can keep a printout of it. You can every day make sure that you go through this cycle once, all right. Next is training. Training means what? Solve the questions, okay. So if you look at, this is one question from your DBT exam. So you get a simple direct question like this, or you can get a question which is something analytical, okay, where they are giving you some options. Now these type of questions are also common for your CSIR, for your GATE BT, for GATE Excel as well. So when you're practicing the question, not only take the previous year question of your DBT, but also go through the questions of CSIR, GATE BT, because the more you go through this question, okay, the more you will be pushing yourself to think about a solution, okay. So that is how you are going to train your brains, okay. You have to train your neurons to act as accordingly. So go through all these previous papers, all right, and try to think in the analytical form. Now focus on the uncommon topics later on. So initially you have focused on the main topics. Now you can focus on the uncommon topics like the bioinformatics, IPR biosafety, environmental biotechnology. Okay, these are the uncommon topics which are not very common to all the other examinations. So for these things, you just have to know basics of it, okay, certain terminology, certain techniques, all right. You may not go into the very details of it. So those kind of information you can definitely Google it out, okay. So whenever you are free, when you don't want to sit with your books, you take your phone, okay, and you just Google out the terminologies which are associated with these topics and jot them down, okay. And lastly is your practice, okay. Practice means what? Revise the topics, solve the previous year's papers, as many as you can. And of course, when you are solving the paper, okay, initially maybe you are going to refer to the paper to see what are the different types of questions which are asked from that topic. But later on, okay, set a time. So before your exam, you should do this. Take up any paper, okay, set the time. So your exam, DBT exam is for 3 hours, right? So you set the time for 3 hours and you try to solve that paper, okay. In that way, what will happen is that solving of paper within the time, that practice will also happen, okay, and that will help you to perform well. Because under relaxed condition, thinking about one solution is something else, and under stressed condition where you have the clock ticking by, okay, under that condition, thinking about a solution is something else, all right. And here this is a place where we actually falter, Okay, we know the answer, but in the examination, because of that tension, because of that nervousness, we failed to give the correct answer, or we mark the wrong one. Though now it has become an online exam, so it has become easier now to mark the options and everything, but still you need to think. Okay, so practice with time, and remember, when you're practicing, practice like you have never won. Okay, and play like you have never lost. That should be the strategy. That should be your way of doing the things. Okay, and lastly is your performance. So when you get the question paper, what you need to do? Okay, first and foremost, see, before your exam, what did I tell you? Sit with a paper and solve it within a time frame, right? So when you're solving in a time frame, you already know which section to give more time and where to give less time. Okay, so immediately when you get your question paper, divide the hours wisely. Now previously, when it was an offline examination, you could roll over all the questions at one glance and then select whichever questions you wanted to answer. But now, since it is an online examination, I would prefer, or I would suggest, that when you're getting a question, just see if you can answer it or not. If you can answer, just give the answer. Okay, if you can't, then mark it for review and go to the next one. Don't try to go through all the question papers, because in that case you will lose your time. All right, and while answering a particular question, do read the options carefully. Okay, because you might get a hint of the answer from the options also, not for all the questions but for certain questions. All right, and lastly, identify the keywords in a question which required to solve and underline them. Okay, because those keywords are going to help you to find the answer. And all of these you're going to do when you are practicing. Okay, so you practice that, and next when you're performing it in the examination hall, you will be able to do it properly. Okay, some extra tips to give off. First and foremost, analyze the X and Y graph. Okay, when you get it, because many a times your question, they are based on what they are asking about, what they have placed on your X and Y axis. Okay, next, pay attention to the unit conversions. Don't get wrong in this unit conversion. All right, do not hurry to answer a question. Read extensively about different topics from various website articles and books in your leisure time. All right, and go through diagrams and glossaries in standard reference books in your leisure time as well. Okay, because these are added benefits that you get. Okay, when you just go through these things. All right, now, so with all the preparations, all of these plannings and everything, I guess you are now ready to give the exam. And when is the exam set tentatively for 2020? It is June 30th, right? So it is high time now. Okay, if you have not yet prepared, you have not sit with your syllabus, you have not done that, then sit with your syllabus, go through it. Okay, and your Biotechnica is going to help you prepare. So in this short notice, Biotechnica has brought you certain courses. Okay, courses which are related to your DBT Biotech entrance exam. You have this DBT BET JRF exam coaching. You have the DBT JRF quick BET revision self-learning coaching. Then you have the DBT mock test series as well. Okay, so when you're preparing, this is a time you opt for these courses and you prepare yourself well. Okay, so that you come out smiling from that examination hall. Okay, so for more references to these courses, you can definitely visit our website. Okay, yourbiotechnica.org, or you can go to stores.biotechnica.org. Okay, there also you will get this details of all these courses. All right, so remember, The successful warrior is the average man with laser-like focus. Alright, so those who are coming out with flying colors, whether it be in the DBT exam or whether it be in your CSIR exam, everyone is an average person. Don't think they are having, extra neurons, extra capability of memorizing. No, but why they have come out with flying colors? Because they had a very sharp focus. They knew what exactly they are doing and they knew how they were doing, okay, and why they were doing. So you also from today onwards build up that focus, okay. So DBT is coming round the corner. Prepare it from day one, and that day one is now, okay, this evening, right? So all of you are going to do it. So let's see if you are going to do it. You can type ETL. Who are eager to do this, who are eager to focus. Don't miss out this opportunity of DBT. Okay. So I see a lot of ETLs coming in. Yes. Good. Very good. So once you are determined to appear for this examination, you can visit these websites, okay, for further details. So you have the BET website, all right, which is your rcb.res.in BET 2020, or you can go through this DBT website, all right, or you can check out our very own Biotechnica website, which is your www.biotechnica.org. So here you are going to get all the informations related to your DBT examination and also any guidance that you need regarding this exam preparation. Okay, you will get it in the Biotechnica website. So do visit these websites, all right, and look for all the informations which are given here. All right. So with this, I come to the end of my discussion. I hope I have been able to clear all your doubts and this session was helpful to you. So I wish you good luck for your examination. Okay, all the best and thank you.
All right. So this was really powerful webinar. I can see that a lot of you have given a very good feedback. How was the webinar so far, guys? Did you enjoy it? Okay, yes, fantastic. Thank you. Thanks a lot. If you enjoyed the webinar, just type fantabulous. Just type FB in your chat box. Fantabulous. If you liked it, just type FB. Fantabulous. Yeah, fantastic. Fantabulous. Thank you. This was really awesome. I really liked the way both of them presented, right? So now that we come to an end to our session today, it is the time for the surprise. So my colleague will right now ping you in chat. One link is there. Please fill your details like what was the feedback? Did you like it? Please rate us based on a scale of 0 to 5, okay, if you like the webinar. Once you have filled the form, automatically you will be redirected to the surprise gift, and also I'm going to send you a personal mail as well. Thanks a lot for attending this webinar, but before you go away, I have a couple of things to share, okay. The first thing is on your screen right now. Trying is the winning formula, okay. Do you remember this famous story you used to hear in your childhood, that there was a tortoise and there was a rabbit here, and they were both competing, and then somebody won the race. I'm sure you remember who won the race. Tortoise won the race, right? Yes. So what exactly it means is someone will always win the race, okay, but you will definitely not win if you do not try. So the first tip or the first formula which I want to give is apply for DBT exam, okay, and start preparing for it now that you have the syllabus, okay. We also have a DBT quick DBT reviser which you can opt for, which will actually help you in winning this exam. Now the next tip which I have is how to be number one. Okay, now it's very simple, as you can see on your screen. Now it's very simple. All you have to do is find the guy who is going to be number one and just score 1 point above him. You are the topper, right? So if that has to happen, this is what you have to do. So now I don't want you to do a Google search and find out who is going to be the CSIR NET topper. Definitely you will not find, or the DBT topper, you will not find that. All I am trying to tell you is you have to use smart work. Okay, you have to utilize smart work and find out loopholes in your preparation and plug those loopholes, and that's how you can be number one. You know who is the topper? Somebody who could plug all the loopholes in his preparation strategy compared to others. As simple as that, right? So that's the thing which I wanted to share. Remember, hard work puts you where good luck can pick you up, okay, or find you. That's the thought. Recently it was put up on Biotechnica. I really loved it. So with these thoughts, I would request you to write DBT JRF exam and prepare it strategically. One more point I wanted to add here is when you go for a future job, in future when you will go for a job and when you will go with your CV and it will be written DBT JRF qualified, also it will be written CSIR NET qualified, the kind of impression you will create on the employer's mind will be, this person is very knowledgeable. He qualified 2 entrance exam just after his MSc, right, or Masters or BTech. So that's the kind of rapport you have to build on your CV which others won't have. Okay, others will simply have only a CSIR while you have CSIR JRF as well as a DBT JRF. So people will really think that you are very, very knowledgeable, and that's where they will put their best bet on. Okay, the world will only salute those who are achievers, right? If you have to be the achiever of tomorrow, then this is the right time. DBT JRF is right here in front of us. After 6 days, the application closes. 18th of May, I guess it should close, but they may extend a few days, like 15 days more, maybe, based on the number of applications people who have applied. But you still have 6 days to apply for this exam. Okay, and if you want any kind of guidance, I'm going to send you a special email just after this webinar, another 15-20 minutes. Okay, I'll just freshen up and then I'll send an email to all of you, and it'll have some very important information about how to prepare for DBT JRF. Okay, and do prepare and study well. All the best. Remember, every adversity is an opportunity. All you have to do is understand it and crack it. Thanks a lot for watching and attending this webinar. It was really a pleasure to interact with all of you. Have a great day ahead. Bye-bye. Good night.